And welcome again all of the people who are right now attending this webinar. And today, so first of all, I should introduce myself. My name is Muzammil Ahmed. I'm scientist 3 at Elixirgen Scientific in Baltimore. And today, we are going to hear two speakers. And today's title is the webinar title is Modeling Neurotoxicity and Neurological Disease Using Functional iPSC-derived Neurons. So we will be hearing two speakers. The first speaker is Dr. Kara L. Gordon, who is a Ph.D. Director Neurobiology, Vala Sciences. And our second speaker is Michal Milrod, who is Scientist 2 at Elixirgen Scientific. So before starting this webinar series, I would like to tell you how this webinar will be proceed. So for question answer, please submit your questions at any time during the webinar using question and answer button on the Zoom application. All the question and answer will be addressed after each speaker's talk, including the slide number will help identify the subject of your questions. We may not be able to address all the questions. Then we will, all the unattended questions will be answered via email. So please keep your name consistent with your registered name so we can find you. So first, I would like to introduce Dr. Michal Milrod. Dr. Michal did her Ph.D. in immunology from the Molecular Biology and Genomics program at the Johns Hopkins University School of Medicine. She did her postdoc at the Johns Hopkins University School of Medicine. She joined Elixirgen Scientific in 2020, and she has over a decade of experience culturing and differentiating human pluripotent stem cells. She specializes in creating consistent, intuitive, and easy to follow user guides. So ultimately, users, clients' time and so that uses mumble time and increase their efficiency so they can focus on achieving their research goals. So let's welcome Dr. Michal Milrod.
Hi, thank you, Muzammil, for the introduction. I would like to introduce all of you today to Elixirgen Scientific. For those of you who are not familiar with us, we were founded in October of 2016 with our headquarters in Baltimore, Maryland. 70% of our research and development team at our headquarters are PhD scientists, and we also have a branch in Japan. Elixirgen Scientific is centered around a core technology known as quick tissue technology, which uses transcription factors to rapidly and efficiently differentiate human iPS into several cell types. Our core technology, the quick tissue technology, is based on identifying genes using a human gene expression correlation matrix to find genes that are highly correlated to the desired cell type. We then produce a cocktail of synthetic transcription factor mRNAs and transfect the pluripotent stem cells with those mRNAs so as to achieve the desired cell type. We optimize the differentiation conditions, and it works quickly and efficiently. This procedure gives us several advantages. It allows us a systematic method of developing our protocols. It results in rapid differentiation. It's robust enough to work with several iPSC lines. Produces cells that are highly functional, do not have a genetic footprint. The protocols themselves are very easy to follow and are similar across multiple cell types. So once you start using our products, you are comfortable using them, branching off if you want to look at a different cell type, etc. Currently, we offer several products and services. We have differentiation reagent kits where you conduct differentiation in your own labs using your iPSC. We also have human iPSC-derived cells that we sell frozen. So we do the differentiation ourselves. We freeze the cells and you can purchase them. We do that from healthy donor lines or from disease cell lines. We have access to the CERM lines, which are over 1,500 unique iPS lines, both contain healthy and disease-derived lines. We sell assay-ready plates. We also have a branch that does differentiation services where we work with you to customize what we offer. If there's a cell type you're interested in that we don't currently offer, if you would like us to differentiate cells using your own IPS lines, if there's an assay that we could perform for you, etc., please reach out. We always love working with our customers. Today, I would like to focus on how our quick tissue products and services can be used to aid your research. Again, our cells function comparably to their in vivo counterparts. They can be used in 2D and 3D models, and thus are very useful for advancing regenerative medicine, for toxicology, for drug efficacy, etc. So neurons that were differentiated with our quick neuron kits display neurites that can have whose length increases over the course of differentiation, and it can be measured. So here we have cholinergic neurons. We're looking at digital phase contrast with the CQ1, and the neurites are labeled in green. And you can see that here's day 10, day 15, day 22. As the course of differentiation progresses, the neurites increase in length all the way up to day 58 here. And this was true in, across several wells, showing consistency in our experiment. We've found it very useful to visualize the neurites using a technique with GFP lentivirus transduction. So we use lentiviral delivery of a fluorescent protein as an easy, non-invasive method to visualize the morphology of the cells while they are living. In this case, we used a commercial, ready-to-use lentiviral vector that has GFP under the human synapsin 1 promoter, which is neuron specific, allowing us to clearly see healthy neurons and differentiate between them and other cells that might be there or other cellular debris. Nearly 100% of the neurons were labeled. We identified an optimal multiplicity of infection, and you can see the fluorescent intensity increases as the MOI increases. Again, this is a very useful technique for live cell imaging to look at neurite outgrowth, to look at neurotoxicity of different compounds, to look at functional assays. It has the advantage of lentiviruses permanently integrating into the genome, and therefore it's very stable expression of the transgene, making it a great tool for long-term studies. So we use this technique for a neurotoxicity study, as an example, with glutamate, which is known to induce neuronal damage through a process referred to as excitotoxicity. And neurite degeneration was seen when 10 micromolars of glutamate was added to the cultures. And it was evidenced by this neurite beating that you can see throughout the culture. And that's actually one of the earlier events in neuronal degeneration, occurs before cell death. So this is a very sensitive assay that allows you to predict toxicity early on in the event. Similarly, we use lentivirus transduction to express a protein for a functional analysis. In this case, we looked at GCAMP. It's a genetically modified calcium indicator, and it uses a fusion of calmodulin, M13 myosin light chain kinase peptide, and GFP, and it fluoresces when it's bound to calcium. So when we expressed a variant of GCAMP in the human iPSC-derived excitatory neurons, we were able to see the neuronal activity and measure it by detecting the intracellular calcium as it fluoresced that was caused by the action potentials. And here you could see waveforms of the calcium oscillations on day 21 and day 29 after thawing of these neurons, and shown in duplicate. And you can see the spontaneous calcium oscillations, which again highlights that this method can be used for long-term monitoring of calcium oscillations, does not really affect the cell viability. And you can detect changes in neuronal activity using a very sensitive assay earlier on than if you were waiting for an endpoint assay. We have also successfully used our cells, the cells derived with our quick neuron technology, with microelectrode arrays. In this case, we are looking at a Maxwell Biosystems Max2 machine, which has 26,400 unique electrodes. It's a non-invasive, label-free method of making a high-resolution activity map of the cells. And we're able to see good synchronous network bursting among the electrodes in all the wells. So here again, you have a lot of electrodes. It measures the number of bursts, the number of spikes per burst, the burst duration, the interburst interval, network activity, and it's a good way of being able to collect data about the function and activity of the cells. So using this system, we measured the action potential. Here, all of these yellow squares are electrodes, and you can see the progression of the action potential. And an action, the conduction velocity of 0.5 indicates a mature neuron, and here we have 0.465, which is very close in the range of a normal mature neuron, showing that the quick neurons are comparable to primary cortical neurons based on the action velocity along their long neurites. Importantly, all the wells in the assay, again, this is a multi-well machine, performed well, performed similarly, highlighting again the consistency of our neurons. We have also looked at the gene expression using RNA-seq and then MEA plate co-culture. So we found here, you can see the IP, this is a principal component analysis, and you can see the IPS are all clustered right here. And the excitatory neurons that we differentiated are here. But when you co-culture the excitatory neurons with primary astrocytes, they become even closer in gene expression, and they are actually very close to the gene expression of human brain, both fetal and adult. And again, we have 2 time points here, day 18 and day 52. So the longer that the cells are in culture, the closer the gene expression profile is to that of human brain. And it really becomes quite similar, especially compared to the initial starting population, displays markers of neurons and microglia and inhibitory neurons, etc. Using these co-cultures on an MEA plate, we were able to test drug responses. So we looked at the response of the cells to known drugs, and we used the MEA to measure spikes in response to 3 different dosages of these known drugs. So we had a negative control. We had a set of drugs that were agonists to different receptors and channels. We had antagonists to other receptors and channels. We also had ion channel blockers. And again, in each case, we tested 3 concentrations. We added the drug, we waited 15 minutes, we checked to measure the spikes, then we added another dosage. We ultimately washed it out, we incubated it and checked that the cells were still functioning afterwards, which they were. We also were able to transport the cells on the MEA plates, and they continued spiking, so this is a very stable system. And so for example, here we had 4AP, which is a known potassium channel blocker, and we expected it to cause excitement of our neurons. And indeed, we see an increase in the number of spikes after the drug was added at increasing concentrations. Here's the baseline, and when we added 30 micromolar, which is our larger concentration, you can see a huge increase in the number of spikes. Similarly, carbamazepine, which is an anti-epileptic drug, is a sodium channel blocker. We expected it to suppress the number of spikes, and indeed, when it was added, you see a decrease in the number of spikes once we got the concentration high enough. If we had a low concentration, we didn't see an effect, but at a higher one, we did. And again, this is the baseline, and when we added 300 micromolars of this drug, which blocks the sodium channel, we did not see the spikes. So again, our cells work really well for testing different drug toxicities. Our most recent product are astrocytes. We now offer iPSC-derived astrocytes, which we're proud to say express astrocyte markers at similar levels to human primary astrocytes. This data here is actually, these are selected genes from a microarray analysis. And if you look at the level of GFAP, of S100 beta, of all these known associated astrocyte genes, you can see that they greatly increase from the iPSC and that they're comparable to the human primary astrocyte. And of course, as the culture continues, you can see these levels increasing as they become astrocytes. Importantly, our astrocytes are also functional, so that when we expose them to 100 micromolars of glutamate, they were able to clear glutamate at level comparable to, again, human primary astrocytes. Again, these are our newest product, and we have developed a suite of products. We are always adding to it. We love to work with you. If there's a cell that you would like that we don't have yet, we currently have excitatory neurons, GABAergic neurons, dopaminergic neurons, cholinergic neurons, sensory neurons, astrocytes, neural precursors, skeletal muscle, and vascular endothelium cells. So at this point, I would like to take any questions anyone has.
And thank you, Mihal. Wonderful talk. And there are several questions. So I will start some questions, and I think one question I can answer here myself. So there is a question, can we get a copy of this webinar? So for your information, yes, our last webinar is also available on YouTube, and this webinar copy will also be available on YouTube after once we finish the webinar. So now I should take more questions on for you, Mihal. So this is the first question is, how important is to adapt the cells to stem fit media before using your product? Is it okay to have them in an MTSR based media and someone is having trouble adapting non-human ES cells to stem fit?
So we found that it's important that the cells are grown as single cells, that they are passaged as single cells. In terms of what media you use, I don't think it requires a big adaptation process. We do have an application protocol for growing, for how we grow our cells using StemFit, which might be helpful to you if you're having trouble with that. But the most important thing is that they're grown as single cells.
Great. And the second question is, have you used your iPSC-derived astrocytes for co-culturing purpose?
So we are, as I said, it's a new product. So we have begun those experiments. We have preliminary data that shows that it works, that it's positive, but it's not definitive yet. We're still testing it and looking at that.
Okay.
That is of course the goal, because it'd be very nice to be able to have both from the same cell line.
So one question is, are these human primary astrocytes or fetal origin? So what is the origin?
Yeah, they are fetal.
They are fetal, okay.
They are human fetal.
One more question, there is very good question, I would say. So on a different note, someone asked, has anyone used this system to create brain organoids, like mini brains, where you have all sorts of different types of neurons and microglia?
So it definitely lends itself well to that. I don't think anyone has as yet formed a complete brain organoid with several different cell types yet. But we would love for you to.
There is one more very interesting question is, how long does it take to make astrocytes with your system?
So again, the longer that you differentiate, the closer it is to the mature astrocyte. So from start to finish, it's about 50 days to get to that mature astrocyte. But if you could stop, at earlier time points, like 28 days, and you just have a kind of a less mature astrocyte. So it depends on what your end purpose is. But again, astrocytes happens to be one of the cell types that takes a very long time. So if you look at other protocols, you will find that most of the protocols are longer than, some of our other cell types, which only take a week.
Okay. So one more question related to media is, have you tested different media for the neuron plus astrocyte co-culture on MEA, and was neurobasal plus the most optimal?
We did, obviously there's a limit to how many tests you could do, but we did optimize and try different, I think different medias or different combinations. But you can contact us, and we can discuss the details of those experiments more.
So there is one more question that is, do you have in vitro model for peripheral nervous system?
For the peripheral nervous system?
Yes.
So again, you could reach out and we can, better address your particular question in terms of your specific needs. But if you go to our website, you could see what we offer, several types of neurons. I don't think we have made specific distinctions between peripheral and central at this point, but we have things in the works and we can work.
I think our sensory neurons can be used for this peripheral nervous system for that question. Yeah. So I think here. So more questions. If you have more and more questions, you can again post for this session for Mihal, and then we will get back to you via email. And till then I think it's a time to welcome our second speaker. So our second speaker is Dr. Kara Elixirgen, and who is the Director of Neurobiology. Dr. Cora Elixirgen, she did her Ph.D. in Neuroscience at University of Iowa. She did her postdoc at University of California, San Diego. She uses Vala's Kinetic image cytometer IC200KIC with high throughput automation and analytics with iPSC-derived cells to measure single cell responses to drugs and in disease conditions. She leads efforts to create iPSC-derived neurons and neuron-astrocyte-microglia co-cultures to model neurological disorders for drug discovery and for neurotoxicity screening. So let's welcome the second speaker, Dr. Kara.
Good morning. Let me share my screen. All right. Thank you so much for having me to be a speaker at this webinar, and thank you everyone who's come to hear us speak. I'd like to tell you how at Vala we're modeling neurotoxicity and neurological disease in iPSC-derived neurons using kinetic image cytometry. So developing drugs for neurological diseases such as neurodegenerative disease has been proven difficult because the models that are often available are animal models such as mice or other cell models that are immortalized or non-CNS cells, and often these cells and animal models don't recapitulate the human phenotypes of disease. Therefore, the models have had low predictivity when it comes to developing drugs for human CNS disease. So the use of induced pluripotent stem cells holds advantages as a cell culture model for CNS disease drug discovery. One of those advantages is that we can find patient or disease-specific lines. We can also derive multiple CNS cell types, such as neurons, astrocytes, and microglia. When the cells are differentiated, they retain the human gene expression or patient and disease-specific cell gene expression profiles of the patient. The neurons that are differentiated from iPSCs also have electrophysiological properties and calcium signaling pathways as do primary neurons, and they form synapses and networks. So here at Vala, we're very interested in using iPSC technology to develop models for neurotoxicity and neurodegeneration. So intracellular and extracellular calcium are important for neuronal signaling and function and influence multiple cell processes. One of the important role of calcium in neuronal signaling is that it can enter neurons due to membrane excitability events through voltage-gated channels or through neurotransmitter-gated channels upon the binding of agonists. Once inside the cell, the influx of calcium affects release of neurotransmitters as at presynaptic terminals, propagating signals between cells. In multiple neurological disease and in aging, there are links to disrupted calcium homeostasis and to neurons dying by excitotoxicity, a calcium-mediated cell death. The influx of calcium into neurons can be detected using fluorescent calcium dyes, and assays can be developed to assess the changes associated with disease, aging, and drug-induced toxicity. So here at Vala, we routinely perform calcium activity assays and iPSC-derived neurons and cardiomyocyte cultures, and we are particularly interested in quantifying single cell activity. This video, which I hope you can see flashing, is a video of fluorescent calcium activity in a culture of Elixirgens mixed neurons in a 384-well dish. The neurons were stained with the calcium dye Rhode 4 and were imaged at 4 frames per second on Vala's IC200 kinetic image cytometer, which is a small footprint imaging device that allows for ultra-fast videography and high throughput analytics of single cell events. The video is being played back at 30 frames per second. Single cell, single transient analysis was performed using Vala's CiteSeer analysis software platform. This analysis is how we extracted the single cell transients for each of the circled cells to the right of the video. As you can see, each cell in this culture exhibits very different behavior. And if you only got the well average shown at the very top of the traces, you would lose a lot of information. With the KIC and the CiteSeer analysis platform, we can extract information such as the event frequency for each cell and the cell peak, cell height, peak height and peak area for every transient of every cell in this video. We also perform routine high content assays using iPSC neurons on our system, allowing us to investigate morphological changes associated with neurotoxicity and neurological disease, alongside our calcium assays. Here we show a fixed endpoint assay for neurite and synapse length, neurite length and synapse count. Here we've stained our cells with pre and post synaptic markers such as SV2 or synapsin and PSD95. And then we've stained with neurite marker, beta tubulin 3 or MAP2, and stained the nuclei. And our analysis software can then distinguish between live and dead nuclei and trace all of the neurites to give us a neurite length. And it can also quantify synapses by determining when a pre and post synapse overlap on a neurite, and that's what we call it a synapse. So here I'll tell you a little story about how we have combined these two methods, the live calcium assays and this fixed endpoint assay on iPSC-derived neurons to develop a model of neurocognitive decline in the context of HIV antiretroviral drugs. So patients who are infected with HIV can develop neurocognitive impairment, although treatment with combined antiretroviral therapy to suppress the virus has lowered the incidence of HIV-associated neurocognitive disorders. This is demonstrated in the figure with the reduction in cognitive impairment risk as the CNS antiretroviral concentration increases. However, 50% of patients who are HIV positive with controlled viral replication still present with HIV-associated neurocognitive disorders. So there are other factors, including toxicity of the antiretroviral drugs themselves, that may be contributing to neurocognitive decline. So here we've plated iPSC-derived neurons in 384-well dishes to investigate the toxicity of some of these ARV drugs using our neurite and synapse assay. We've used 4 of the currently prescribed drugs for a 7-day treatment at 3 doses each. First, we can see that the highest dose of 3 of the 4 drugs decreases the neuronal viability. Additionally, one or more concentrations of each of the 4 drugs reduces the synapse count in these iPSC-derived neurons. Next, when we look at neurites, we've shown here images of the beta-2 staining of the neurites on the cells after the drug treatment at the highest dose. And you can visibly see that many of these drugs reduce the neurite length. And we can see after quantification that that is true. And 3 of the 4 drugs at the highest dose reduce the neurite length. So combining all of these 3 measures, we can see that the drug emtricitabine or FTC shows the least toxic effects in a neuronal model, and the drug elvitegravir or EVG shows the most pronounced toxic effects. Next, we assess the function by live calcium imaging using our kinetic image cytometer. Here we've used a 7-day treatment of the highest dose only, both with the drugs alone and in combinations. So the antiretroviral drugs belong to different classes with different mechanisms, and they are prescribed in combination to attack viral suppression in multiple ways. So we've used combinations that are used in real life. So here we see that both dolutegravir and elvitegravir were toxic to the cells and reduced the percent of active cells in culture, both alone and in their combination therapy. Elbitegrivir, again, was the most toxic. Additionally, it reduced event frequency and the mean peak amplitude of the calcium transients. And it is definitely more toxic in combination than it was alone. Next, we wanted to see if we could protect the neurons against ARB toxicity by adding microglia. So these are images of neuronal co-cultures alone or neuronal co-cultures containing microglia that we produced here at Vala Sciences. This is images for the DMSO control. You can see visually that the lower image with the microglia in the co-culture have more dense neurites, suggesting that there is a nurturing effect of the microglia in the culture. We treated again for 7 days with the antiretrovirals and assessed the calcium activity in both the neuron and microglia co-cultures. The graph is displaying relative activity, so each of the treatment conditions has been normalized to its DMSO control for its appropriate culture condition. And you can see that addition of the microglia to the cultures attenuates the toxicity that is seen with elbitegrivir and in the elbitegrivir combination. Although it is still toxic, it's just less toxic to the percent activity in the cells. So this would suggest that microglia are protective and that our research would also benefit from having toxicity studies in a co-culture model with neurons, microglia, and astrocytes as well for future studies. So actually we're currently working towards making triculture models. So our main goal would be to have iPSC-derived neurons, microglia, and astrocytes, as well as having an isogenic triculture model, which means each of these 3 cell types would be derived from the same iPSCs in our lab. So we're really excited when Elixirgen came out with their iPSC-derived astrocyte kit, because then that allowed us the opportunity to make these 3 cell types more easily and quickly in our lab. So here we are currently working out our co-culture with Elixirgen's quick neuron excitatory neurons and their quick glia astrocytes. And so this is actually a bright field picture that I took earlier this week of these 2 cell types in culture. And then we will just add our microglia, and then we'll see how the cultures act and how their function looks and how they act with the antiretrovirals. So we're really excited to move forward with these cell cultures. So HIV positive pregnant mothers will take antiretroviral treatments while they are pregnant. So we are also interested in establishing a developmental model for ARV toxicity. To do this, we've acquired Elixirgen Scientific's neural precursor cells, and we're setting up our studies to look at proliferation. So here we've used a kit called the Clickit EDU cell proliferation assay from Thermo Fisher, where you add an EDU that will incorporate into your cells during the time of incubation, and then you use this Clickit chemistry that can have a fluorescent endpoint at the end to identify the cells that underwent proliferation. So this image shows the Elixirgen's NPCs stained either with DAPI, the nuclear marker, and some of them with the Clickit EDU fluorescent chemistry to mark the proliferative cells. And just to set up the model, we've utilized 2 drugs that can modulate toxicity and proliferation in the cells. So here you can see that the drug A that we used had a significant effect on toxicity. It killed the cells, more of the cells in the well, but it did not have an effect on proliferation. And drug B that we used did not have an effect on toxicity, but did increase proliferation. So now we feel we've set up the model and we are ready to treat these cells with our antiretrovirals and see how they act in this developmental model. So we're really excited about using co-cultures here at Vala because we think it will be able to model the CNS environment more closely. So we've been doing a lot of work trying to work with co-cultures and to determine methods to differentiate activity from different cell types in the co-culture. So I wanted to share with you a way that we have been working towards differentiating calcium activity from different cells that are cultured together. So in this case, I've cultured excitatory neurons with microglia, and we've performed live cell imaging assays first, as you can see with the ROD4 and DAPI in the top left corner. After we perform the live calcium imaging assays, we fix the cells and then perform immunostaining for cell biomarkers, beta-tubulin 3 for neurons and IBA1 for microglia. So we've developed an algorithm with our CiteSeer software analysis program that will automatically overlay these two images. And so now we can have our calcium activity overlaid with an identifier of cell type, as you can see in the merged image below. So here we have two cells that are circled or circled in red with red squares. And these are neurons, as you can see by the beta-tubulin staining, and you can see their associated calcium activity to the right. And there are two cells that are circled in yellow, and these are microglia based on their IBA1 staining, and they have calcium activity as well. So you can see that the calcium activity of the microglia and the neurons is quite different. The neurons are much smaller, much faster. The microglia have very, large, sort of longer activities. And if in doing single cell analysis, we were to combine these, it might make our data a little bit more messy because they're so different and we may lose some of the changes. So it's important to us to be able to differentiate these two activities. So we've used this method here in an Alzheimer's disease model where we're trying to look at hyperactivity in Alzheimer's disease. We've treated these co-cultures for 48 hours with amyloid beta 1 through 42 fragments, which are the toxic fragment associated with Alzheimer's disease. And then we've done single cell analysis on the live calcium imaging and pulled out the neuronal calcium transients only. And here you can see, if we look at the percent of active cells in these cultures, that as the concentration of beta amyloid 1 through 42 increases, so do the percent of active cells in the culture. So we've been able to see hyperactivity due to beta amyloid 1 through 42 treatment. So we're very interested also in creating neurons and other cell types from disease-linked mutations, and we're actively working towards this. So one of the things that we have done is that we've created an isogenic iPSC line with ApoE33, or the Alzheimer's disease associated ApoE44 genotype. Here we've used Elixirgen Scientific's excitatory neuron kit to differentiate these ApoE33 and ApoE44 iPSCs into neurons, which we have cultured with human primary astrocytes. I hope that you can see the videos of the flashing cells in the center of the screen, and you can see that we've stained them with beta-tubulin and GFAP for the astrocytes as well. So we were able to make very beautiful functional cultures from each of these two lines using Elixirgen's excitatory neuron kit. And you can see this by the flashing in the videos and with the single cell calcium traces that we got from our CiteSeer analysis. To the right, you can see that the many of the cells are synchronously firing, spontaneously and synchronously firing in both of these cultures, as shown in the parts that are underlined by red. So our goal would be to evaluate both these two lines and other disease-linked lines for phenotypes that we can use to create assays for drug development. We are also interested in synchronicity of cultures because this can be a model for drug or diseases such as seizure. So here we've used the drug 4-aminopyridine, potassium channel blocker that you saw in the previous presentation to increase the activity and synchronization of firing in the cultures. This also does allow us to show how our kinetic imaging system can capture fast calcium, changes in calcium activity from fast-acting drugs. As the previous data that I showed you were 7-day treatments, here we can add drugs to the wells even in the middle of a scan with our fluidics capabilities. Although in this case, I have used 2 different wells as my DMSO and 4AP treatments. So in this case, we've used healthy iPSCs with Elixirgen's excitatory neuron kit, again, cultured with human astrocytes. And shown are the fluorescent calcium videos of the DMSO and 4AP treatments at 30 micromolar, and the single cell calcium activity traces to the right. So the DMSO control, you can see these cells have many calcium events, they're just not synchronized. However, after the addition of 4AP, the cultures both increased in activity and increased in synchronicity. So here we've been able to quantify the event frequency of each of the events from each of the cells in these cultures. And you can see that the addition of 4AP has significantly increased the event frequency in these neurons. Additionally, one of the analysis that we can do with our CiteSeer platform is to look at cells with correlated activity. So I've pulled out a large number of cells here to the very right bottom corner with correlated activity, and you can see that these are the cells that are synchronized due to the increase in firing with the 4AP. So here at Vala, we've really found that iPSC systems are very powerful, and we've been using iPSC-derived neuronal cells and astrocytes and microglia to develop models for neurotoxicity and efficacy for drug development and to study neurological disease. And as you can see, we're taking advantage of many of these benefits that I described earlier, using disease-linked lines, deriving multiple cell types for CNS cell types for our experiments, and using the calcium signaling pathways and the ability of these cultures to form synapses and neurites. And so if you're interested in learning more about what we're doing at Vala Sciences, please visit our website. And again, thank you so much to Elixirgen Scientific for allowing me to participate today. And now we can take some questions.
Excellent talk, Dr. Kara. And there are questions for you. So I'm going to read a few questions.
Okay.
So first is, someone is curious about what medium you use to culture mixed Elixirgen-derived neuron cultures.
Like if for the, I guess for all of these, for both these where I had the Elixirgen excitatory neurons with human astrocytes, or so far the Elixirgen neurons with the iPSC-derived Elixirgen astrocytes, I'm just using NeuroBasal Plus, and I haven't tried anything else. And then in the other experiments that don't involve Elixirgen neurons, I generally use just a very general basic co-culture media, kind of DMEM neurobasal that can be found in the literature.
And second question is, do you think it is possible to use Elixirgen products to create a brain organoids?
I would think it would be. I've never actually created a brain organoid myself, so I don't have a super lot of experience there, but I cannot see why not.
And the third question is, are there any issues with allogenic microglia in a mixed culture situation? In the mix, yeah.
So we're currently doing this mixed culture with Elixirgen quick neurons and quick glia, and so right now there aren't even today as I speak microglia in this culture, but there will be next week, and then we'll see how that goes. So far, I have done cultures with CDI astrocytes and neurons and our microglia as well. So I have definitely done 3 together.
And someone is saying really nice talk, and a few questions from the attendee is are the microglia iPSC derived? These microglia are iPSC derived?
Yes, the microglia that I've used are iPSC derived.
Okay, the second question from the same person is, were the microglia added to an existing culture of neurons?
Yes, I usually add them a few days after plating the neurons. I think I have tried adding them at the very same time, and that's also fine. But I guess sometimes we just decided to, especially if the media of the media that the neurons come with is different than the media that I grow them in co-culture with. I guess I find that it's maybe nice to culture the neurons for a few days just to give them some of the good nutrients in the media that they come with.
And how long were the cells co-cultured together to see the improvement of neuronal health?
14 days.
14 days, okay. And there is one question, is the kinetic image cytometry be applicable for microfluidic devices?
That is a good question. As long as because we can load different types of well plates or different types of slides into into the base to do the imaging. So as long as whatever is in the device isn't going to obscure the imaging, it should be fine.
Mhm. And yeah. So someone asked that you presented a mix of iPSC-derived neurons as iPSC-derived cells, excitatory neurons, astrocytes, and microglia that are either commercially available or differentiated in-house. Can you comment on the performance or differences between them?
Sure. Yeah. So all the microglia that I've shown today have been differentiated in-house, but I've used different types of neurons and microglia, or sorry, neurons and astrocytes. And if you only need, say, wild type neurons, then it's easy to get the commercially available ones. But when we want to make, a disease line, then it's really nice to have the Elixirgen kit because I can actually make that in-house. I guess the way like the CDI neurons come more mature, which sometimes has been nice because we can plate them and they maybe stay a little bit more nicely, but I've gotten much better activity actually out of the Elixirgen cells than in my hands I have from the CDI cells.
So someone asked, what differentiation method do you use to make microglia? Is it based on the Abbott's protocol?
I would say no, and they can definitely give you my contact and we can talk more about this offline.
Okay, that's okay.
Yeah.
And someone also like to know, are the ApoE3 and ApoE4 iPSC lines available for sale? Commercially available?
We've made them in-house and we aren't currently selling them, but we could certainly do screening with them if you're interested. That's another, you could contact us and we can discuss.
Okay, that's great. And maybe one or two more questions. So how many single cells can you take measurements of actually in one well with the cytometer?
That's a good question. So in a field, we get about 1/10 of a 384 well when we use a 20x. And that can usually be anywhere between 100 to 200 neurons, and we can get a reading from each of those cells in the field. This is a cropped field that you're seeing right here, so this maybe only shows 50 to 100. And then when we're doing the neurite outgrowth, we can often take 9 fields, which is almost the entire well, and we get about 2,000 neurons for that. You can get a lot of data.
Yeah, okay. So one more question is that where you co-cultured neurons and microglia, have you actually quantified the neurites there in that? Yeah.
Yes, we did quantify the neurites in this slide right here, and we were able to see that the microglia significantly increased the neurite length when they were co-cultured in there for 14 days. So it's a visible difference.
So I think, yes, we have asked enough questions to you, and I think a lot of questions were asked. And again, so if viewers have more questions, they can paste the questions and we will get back to you via emails. And thank you so much for attending this session. And there are a few more things I need to tell you. So just please wait. So yeah, there will be a next webinar where there will be 3 speakers, and this webinar will be about tools to study neurodegeneration, iPSC-derived neurons, nerve organoids, and engineered human synaptogenic beads. So this webinar will be on December 10th, 2021, 9 to 10 a.m. East time. And here's the barcode. So for easy registration, please scan this barcode, and the questionnaire will be available after 24 hours of posting this advertisement. So for easy registration, you can scan this barcode, and this will be available on social media platform too. So if you are not able to do right now, you can do it later. And this video again after this, because someone asked also, so this webinar video will be available on YouTube, and before this one, the first webinar is also available, and we would like to do with all our webinars to post on YouTube so that you can revisit, and you can, if you have any questions, you can get back to us. So I would like to request also to visit our website, and if you have any questions, and we will be great to know how we can help you in your research. So please. I would like to welcome again for the next one in advance. So please scan this barcode. And thank you so much for attending this webinar, and enjoy your day. Thank you so much.